Welcome back. Today's topic is Chapter 5, the plasma membrane. So the plasma membrane was discovered in the 1890s. A cell's plasma membrane is what defines the cell and outlines the borders of that cell. It's also what regulates the passage of materials. It can exclude some substances from making it into or out of the cell, while it can take others in. It's also capable of participating in chemical reactions. The cells can excrete some substances and signal other cells. The plasma membrane was identified in the 1890s, but its chemical components weren't discovered until 1915. In 1915, they were able to identify lipids and proteins. However, it wasn't until 1935 that two scientists named Hugh Davidson and James Donnelly proposed the plasma membrane structure. They theorized that the plasma membrane structure resembled that of a sandwich and made the analogy of proteins to bread and lipids to filling. A little bit later in the 1950s, they were able to tell that the plasma membrane's core consisted of a double layer rather than a single layer. But then in 1972, S.J. Singer and G.L. Nicholson proposed a new model that better explained how that plasma membrane actually functions. This explanation was called the fluid mosaic model. The fluid mosaic model describes the plasma membrane structure as a mosaic of components that gives the membrane a fluid character. They were able to see that there were two layers of phospholipids with other proteins and lipids embedded within the layer. So since the plasma membrane consists of a phospholipid bilayer, I just want to remind you what a phospholipid is. So that is an amphiphilic molecule, meaning that it has a hydrophobic region and a hydrophilic region. So in the phospholipid, it has a hydrophilic head that is made up of a phosphate and a glycerol. So that's the water-loving portion of the lipid, hydrophilic. And then it also has a hydrophobic tail consisting of saturated fats and unsaturated fats. And that's the portion that forms the inner sort of layer of the phospholipid bilayer, with the polar heads facing outwards towards the outside of the cell and then the inside of the cell. The membrane's mosaic character helps to illustrate its nature. Both the proteins and lipids exist in the membrane as a separate but loosely attached molecule that float and move somewhat with respect to one another. Now, the membrane is not able to expand and contract like a balloon. It's a fairly rigid structure that can burst if penetrated or if the cell takes in too much water. A very fine needle can easily penetrate a plasma membrane without causing it to burst. This is because the membrane will flow and self-heal when the needle is removed. There are a few things that can influence the fluidity of the plasma membrane. The first is whether or not it's composed of saturated fatty acids or unsaturated fatty acids. If it's composed of unsaturated fatty acids, the membrane will be more fluid. This is because the double bonds in unsaturated fatty acids, or the kinks, will keep them from packing tightly. Now, the other thing that can influence fluidity is temperature. As with many other liquids, increasing the temperature and making it warmer increases the fluidity of the liquid. So is the same with the plasma membrane. The warmer it is, the more fluid the membrane will be. Within the phospholipid bilayer, you'll find different types of proteins. Some of the proteins may be embedded within the bilayer. These are called integral or intrinsic proteins. Integral proteins integrate completely into the membrane structure, and their hydrophobic nature interacts with the phospholipid's hydrophobic region. Some of these proteins will span only part of the membrane and associate with a single layer, while others will stretch clear from side to side. These are called transmembrane proteins. You can see a transmembrane protein here. It stretches all the way through the membrane. These transmembrane proteins will have a hydrophilic region and one or several hydrophobic regions. This arrangement of the protein will orient it alongside with the phospholipids, with the protein's hydrophobic region adjacent to the phospholipids' tails and the protein's hydrophilic region or regions protruding from the membrane and in contact with the cytosol or extracellular fluid. Peripheral proteins, or extrinsic proteins, are those proteins that are associated with the surface of the bilayer, meaning that they're on the membrane's exterior or interior surfaces. They can be attached to either an integral protein or to a phospholipid. Both peripheral proteins and integral proteins may serve as enzymes or as structural attachments for cytoskeleton's fibers. They may also be part of the cell's recognition sites, which we refer to as cell-specific proteins. The body will recognize its own proteins and will attack foreign proteins associated with invasive pathogens. Transmembrane proteins may appear in one of two forms. They may have one or more alpha helixes that span the membrane, like examples 1 and 2 in this image, or they may have beta sheets that span the membrane, like example 3. The alpha helix is a shape produced by a certain chain of amino acids. The interactions between the amino acids next to each other make a downward and inward bend, creating a structure similar to a spiral staircase. Alpha helixes tend to be nonpolar, which gives them a distinct advantage to staying bound within the hydrophobic tail region of the membrane. An integral protein may only have one region of alpha helix, as shown in the number 1 image here. Or they may be a polytopic alpha helical protein, which has multiple helices traveling through the membrane. A beta sheet is a complexly folded chain of amino acids which forms a flattened rigid sheet. When many beta sheets extend through the membrane, creating a pore, the structure is called a beta barrel. The outsides of the beta sheets have hydrophobic residues, and the integral protein can be locked into the plasma membrane. Like the transmembrane alpha helix, the beta barrel requires the correct sequence of amino acids for the integral protein to maintain contact with the membrane. This slide is just for practice. You will see something like this on your exam, so be sure to take a look at it. Membrane proteins have many important functions. They may be what's called a transporter, which brings molecules from outside of the cell into the cell, or vice versa. They may also be an enzyme, which, as you know, enzymes catalyze reactions. They will bind to a substrate and then release a different product. Membrane proteins may also be cell surface receptors. Cell surface receptors are membrane-anchored proteins that bind to ligands on the surface of the cell and then release a signal to the cell. Another type of membrane protein is a cell surface identity marker. Cell surface identity markers are used to classify cells according to that marker, so that other cells know exactly what type of cell that one is. Membrane proteins may also be involved in cell-to-cell adhesion. They can bind cells to one another. They're also capable of attaching to the cytoskeleton, which gives more structure to the cell. In addition to lipids and proteins, the plasma membrane's third major component is a carbohydrate. Carbohydrates are always on the cell's exterior surface and are bound to either proteins or to lipids, forming glycoproteins and glycolipids respectively. These carbohydrates will form specialized sites on the cell surface that allow cells to recognize one another. This recognition function is really important to cells because it allows the immune system to differentiate between body cells and foreign cells or tissues. Now, similar glycoprotein and glycolipids may appear on the surfaces of viruses and may change frequently, which prevents the immune system from recognizing and attacking them. In animal cells, you will also find cholesterol within the plasma membrane. Cholesterol can fit into spaces in between phospholipids and prevent water-soluble molecules from diffusing across the membrane. This slide just shows you another look at the plasma membrane and how it's structured. And if you want to learn a little bit more about the plasma membrane, go ahead and check out this fluid mosaic video, and it will definitely help you piece these things together. Now, once you're a little bit more familiar with the membrane, I encourage you to draw it and label the components. Now, a simple illustration can look just like this. I just want you to be familiar with what those components are and where to find them within the membrane. It'll also be valuable to label which side is the inside of the cell and which side is the outside of the cell. So earlier I said that the plasma membrane was selectively permeable. Now, selectively permeable means that the cell will allow some substances to pass through, but not others. So what is allowed to pass through and how does it do it? Well, biological membranes are permeable to small, nonpolar hydrophobic substances. This is because plasma membranes are amphiphilic, meaning they have a hydrophobic and a hydrophilic region. Nonpolar and lipid-soluble materials with a low molecular weight can easily slip through the membrane's hydrophobic lipid core. These are substances such as fat-soluble vitamins like A, D, E, and K, as well as oxygen and carbon dioxide. Polar substances like sodium, potassium, and calcium ions are not able to slip through the plasma membrane because they are charged. So if some substances can slip in and out of the cell through the plasma membrane, then what determines where they're moving or what direction they're going? Well, this has to do with the law of entropy. Hopefully you remember what entropy is. Entropy is the amount of disorder within a system. So a substance or molecule or any type of matter will constantly be trying to move from a less stable state to a more stable state. So from more organized to more chaotic. So what we see in this example is someone dropping dye into a bunch of water. Well, when that dye initially hits the water, it stays relatively close together so that you have a small dark red drop of dye. But as time goes by, that dye tends to spread out and diffuse across the rest of the water. So it is going from a more ordered state, a pure dye that is contained, to a less ordered state as it spreads out and diffuses across the water. Diffusion defined as the movement of a particle from a high concentration to a low concentration, or in other words, down the concentration gradient. And this is the same way that it works when passing through a cell or through a selectively permeable membrane. If there is a high concentration of a particle on the outside of the cell, then it will move into the cell. If there is a really high concentration of that particle within the cell, then it will move out of the cell. Now, diffusion will continue until the concentration is the same on both sides of the membrane. Diffusion is a type of passive transport that allows diffusion across a selectively permeable membrane, such as the plasma membrane. Passive transport is a type of transport across the membrane that does not require the input of energy. The opposite of this would be active transport, which does require the input of energy. Now, there are two types that we're going to talk about. There's solute diffusion and solvent diffusion. Now, if you can think about the characteristics of a solution, you have to have a solute, which is something that will dissolve in a solution, and you have to have a solvent, which is the substance that will be doing the dissolving. Typically, we use something like water, or water mixed with something else to be a solvent. So the passive transport of the solute involves small particles moving through a solvent in order to reach an equilibrium. Now this example shows dialysis tubing with a mixture inside of the dialysis membrane. Now, over time, the smaller particles are going to diffuse outside of that membrane until there is an equal concentration within the membrane and outside of the membrane. The larger particles are not permeable to this membrane, meaning that they cannot travel outside of that membrane, so all of those will remain inside that dialysis tubing. Another type of passive transport is called facilitated diffusion or facilitated transport. With this type of movement, materials are able to diffuse across the plasma membrane with the help of membrane proteins. So in order for this to work without the expenditure of energy, a concentration gradient has to exist, meaning that there is a heavy concentration of the molecule moving on one side and a low concentration on the other. Now, the types of molecules that are going to be moving through these proteins are polar. So the protein actually shields these materials from the membrane's repulsive forces and allows them to diffuse into the cell or out of the cell. The transported material will first attach to a protein or a glycoprotein receptor on the plasma membrane's exterior surface. So this will allow the removal of the material from the extracellular fluid that the cell needs. The substances then pass to specific integral proteins that facilitate their passage. Now there are a couple of different types of proteins that allow for facilitated diffusion. The first is called a transport protein. Now, a transport protein is a type of integral protein that functions as either a channel for a material or a carrier. In both cases, they are transmembrane proteins and are substance-specific. Channel proteins have a hydrophilic channel through their core, which provides a hydrated opening through the membrane layers for substances to move through. Now, channel proteins can either be open at all times or they're what we call gated, which means that they can close depending on certain circumstances. When a particular ion attaches to the channel protein, it may control the opening. Okay, so when we're talking about gated channels, there are a few different ways in which a channel can open or close, and in which direction those molecules will be moving. Ligand-gated ion channels bind to neurotransmitters and open in response to ligand binding. Another type would be voltage-gated channels. Voltage-gated channels are activated by changes in the electrical membrane potential near that channel. Mechanically gated channels open because of the physical distortion of a cell membrane. In other words, they are associated with a sense of touch. And then there are channels that remain open. The movement of molecules across those channels will be dependent on the concentration gradient on either side of the plasma membrane. Another type of protein involved in facilitated diffusion is called a carrier protein. A carrier protein is a protein that binds to a substance which triggers a change in its shape and then allows the molecule to move across the plasma membrane. This is a process that requires a concentration difference across the membrane. And these proteins can not only move material into the cell, but depending on the concentration gradient, can move it outside of the cell as well. They are a single substance specific protein, and because of the change in shape that is required, they move at a much slower rate than channel proteins. Another type of diffusion that involves the movement of the solvent rather than the solute is called osmosis. Osmosis is the movement of water through a semi-permeable membrane according to the water's concentration gradient across the membrane. Before scientists discovered that there was actually a protein that moved water across the membrane, they thought that despite water being polar, it must be able to move through the membrane because of its small size. But in 1992, Peter Auger and his team discovered aquaporins. Aquaporins are channel proteins that allow water to rapidly move in and out of the cell. Peter Auger discovered this while researching the Rh blood group antigen. They isolated the Rh molecule, but then found a second molecule. At first, they just thought that it must be a piece of the Rh molecule, or a contaminant, but it turned out to be something that had yet to be discovered, which we now know is an aquaporin. Go ahead and take a look at this video on aquaporins just to see a little bit more about how they look and how they function. Now, the movement of water across a membrane is determined by the tonicity, or in other words, the concentration of the solutions on both sides of the membrane. If the solution is the same on both sides, we call this an isotonic solution. Now, this example shows an isotonic solution that is made up of 95% water and 5% sodium chloride on both sides of this membrane. So because of that, no osmosis will occur. There's no need for anything to move because they're equal on both sides. Now, if the solution is not the same on both sides of the membrane, then the side that has a higher concentration of solute will be called a hypertonic solution. And the side that has a lower concentration of solutes, or in other words, the molecules, the material that is not the water, is called a hypotonic solution. So in this first example, we see that there's about the same amount of water on both sides of the membrane, but there's a different concentration of glucose, or the solute, on both sides of the membrane. On the left, we have a hypotonic solution at 5% glucose, and on the right, we have a hypertonic solution of 15% glucose. So the water will move to the area that has a higher concentration, and when it does so, even though it looks like there is more water on this right side than the left, the ratio of solvent to solute is the same. In this example, we see the same starting formula, but this membrane is permeable to both the water and the solute. So in this case, both the solvent and the solute will move across the membrane until they create an equal proportion of both on both sides of the membrane. So in this example, we have 0.03 moles of sucrose inside our mock cell and 0.01 moles of sucrose on the outside of the cell. So the environment would be the hypotonic portion of the solution because it has a lower concentration of solute. And if osmosis were going to occur, which it would in this situation, which direction would it move? Well, the water from the outside of the environment would move into the cell because the cell has a higher concentration of solute and a lower concentration of solvent. When water moves across the membrane, a pressure is created called osmotic pressure. And the pressure will increase where the solute concentration is higher. So in this example here, we can see that there is a higher concentration of molecules on this left side. So there will be a higher amount of osmotic pressure on this left side of the membrane. Now, changes in osmotic pressure can produce a change in the type of cells that we see. A hypertonic cell, as shown in this left cell, will lose all of its water and shrivel. In an isotonic cell, you'll see a plump cell, but one that's neither bursting nor shriveling. And then in a hypotonic solution, the cells will actually start to rupture. They will swell so much because so much water has gone into the cell that they can rupture. Plant cells are similar, but also slightly different. A hypertonic plant cell will be completely wilted. The plant will fall over just limp because all of the water has left the cell and the cell body will shrink away from the cell wall. A plant cell in an isotonic solution will be slightly flaccid because water is moving in and out at the same rate. plants function best in a hypotonic solution, where they have turgor pressure that is very high and keeps the plant upright. Okay, so here is where I'm going to offer you a little bit of extra credit. If you send me an email with a subject titled Practice 4 and answer these 2 questions, I'll give you 5 bonus points or extra credit points. Now we're going to talk about active transport. Active transport is the movement of a substance from a place of a lower concentration to a place of a higher concentration with the use of cellular energy. Now, these are not spontaneous reactions, they are endergonic reactions, meaning that they gain energy through the course of a reaction. Now, the type of protein that we use for active transport is called a transporter protein. Now, these proteins may carry one molecular ion in one direction, such as called a uniporter. Other transporter proteins may be able to carry 2 different molecules or ions in the same direction. These are called symporters. The third type of transporter protein is called an antiporter, which is able to carry 2 different molecules or ions, but in opposite directions. Electrogenic pumps are protein carriers that use energy to concentrate ions on one side of the membrane, which generates voltage across that membrane. Now, this voltage potential is called membrane potential. For example, the sodium-potassium pump is the major electrogenic pump in animals. The sodium-potassium pump moves potassium ions into the cell while moving sodium ions out of the cell at the same time. The rate of exchange is 3 sodium ions for every 2 potassium ions moved. The process of moving these ions consists of 6 steps. Initially, there will be an enzyme oriented towards the cell's interior, which has a high affinity for sodium ions. 3 sodium ions will bind to the protein. Then the protein carrier will hydrolyze ATP and a low-energy phosphate group will attach to it. Then, as a result, the carrier will change shape and reorient itself towards the membrane's exterior. The protein's affinity for sodium decreases and the 3 sodium ions leave the carrier. This shape change increases the carrier's affinity for potassium ions, and 2 such ions will attach to the protein. Subsequently, the low-energy phosphate group will detach from the carrier. And with the phosphate group removed and the potassium ions attached, the carrier protein will reposition itself towards the cell's interior. Then the carrier protein in its new configuration has decreased its affinity for potassium, and the 2 ions will move into the cytoplasm. Then the protein will now have a higher affinity for sodium ions, and the process will start over. Now, as a result of this transaction, several things have happened. There will be more sodium ions on the outside of the cell than on the inside, and more potassium ions inside than out. And for every 3 sodium ions that move out, 2 potassium ions moved in, which results in the interior being slightly more negative relative to the exterior. The difference in charge is important for creating the conditions necessary for a secondary process. So a few important things to remember about the sodium-potassium pump is that it's a transmembrane protein that is expressed in virtually all cells of higher organisms. There are between 800,000 and 30 million pumps on the surface of a cell. Now, the action that takes place with the sodium-potassium pump creates both an electrical and a chemical gradient across the plasma membrane called a resting membrane potential, which is necessary for secondary functions. Without that membrane potential, nerve and muscle cells could not undergo excitation. It is also important for several protein carriers, including those that import glucose, amino acids, and other nutrients. They depend on the export of sodium ions from the cell. The translocation of sodium from one side of the epithelium to the other side creates an osmotic gradient that drives absorption of water in the kidney. Now, coupled transport is the secondary function that I was talking about, and may also be called secondary active transport or co-transport. Secondary active transport uses ATP indirectly as it brings sodium ions and potentially other compounds into the cell. A sodium ion concentration builds outside of the plasma membrane because of the primary active transport process. And this is what creates the electrochemical gradient. If a channel protein exists and is open, the sodium ions will pull through that membrane. This movement transports other substances that can attach themselves to the transport protein through the membrane. Many amino acids as well as glucose enter a cell this way. The secondary process also stores high-energy hydrogen ions in the mitochondria of plant and animal cells in order to produce ATP. The potential energy that accumulates in the stored hydrogen ions translates into kinetic energy, and the ions surge through the channel protein ATP synthase, and that energy then converts ADP into ATP. And this is also an example of a symporter. So that's a type of protein that moves two molecules in the same direction. Now this slide just shows exactly what we talked about. As two sodium ions enter the cell, glucose is able to hitch a ride, so to speak, against its concentration gradient into the cell. So we've been talking mainly about molecules moving one or two at a time into a cell. Now we're going to talk about how movement of bulk molecules or really large molecules move into and out of the cell. Exocytosis is the movement of bulk or large substances out of the cell. Now this type of movement also requires energy. Waste material from inside the cell will be enveloped in a membrane and will fuse with the plasma membrane's interior. This fusion opens the membranous envelope on the cell's exterior, and the waste material expels into the extracellular space. Now the opposite of exocytosis is endocytosis, or the movement of large molecules into the cell. Now there are a couple of different types of endocytosis, but all of them share the same common characteristic of moving material into the cell. First, the cell's plasma membrane will invaginate and form a pocket around the target particle. Then the pocket will pinch off and result in the particle being contained in its own vesicle within inside the cell. Now, phagocytosis is the process in which cells take in large particles such as other cells. An example of this would be when microorganisms invade the human body, and a type of red blood cell called a neutrophil will remove the invaders. And they do so through this process. They will surround and engulf the microorganism and then destroy it. Penocytosis is the movement of fluid into a cell. So it's the same process, but it's only moving fluid in this case. These types of vesicles will be much smaller than those involved with phagocytosis. Now, another type of endocytosis is called receptor-mediated endocytosis. In this case, specific molecules will be taken in after they bind to a receptor. Now, the process for receptor-mediated endocytosis is basically the same as the other types of endocytosis, except first, the receptor will bind, and then the plasma membrane will invaginate and engulf the particle and bring it into the cell. Alright, to finish up this chapter, just take a look at these two videos right here, and that will conclude Chapter 5.